Hello, this is a summary video of Professor Fletcher's lecture. Um, this video is meant to aid you in understanding the main concepts in her lecture, but I highly recommend still attending the lecture and watching the lecture recordings. In terms of learning outcomes, I would first like to go over the types of gene defects before I go into the gene therapies, because we need to know what's gone wrong before we can suggest something to make it right. Now, in terms of the three main parts to this summary series, I will first go over the antisense therapy, and in this there are three components. So first we have transcript degradation, the translational blockade, and finally the altering mRNA splicing, which you can see here. I've had another subset because this is what Professor Fletcher's research is mainly on, which is exon 51, which helps with DMD. Now, you'll notice that there's a little recap portion here. This section is supposed to help you to remember the difference between DMD and the less severe phenotype BMD, and Dr. Fagao covered this more in depth in his lecture. And he also briefly goes over the other gene therapies that we use for DMD. So without further ado, let's go. Okay, this looks like a really busy slide, but I've tried to, if you just take a look at the colors, this has a whole, more or less standalone, and these two I've grouped together because their effect is aborted translation, and with these last two, they are, somewhat related, with the triple repeats in the coding region, like Huntington's, which is CAG, and you have in the non-coding region, which is your Fragile X, which is your CGG, and the contraction here is referring to this very long disease name, which I'm not going to read out. But in Professor Fletcher's slide, she did say that this is an illustrative example. So yeah, it's just good to, quite fascinating actually, if you do a quick search on it. Alright, so in terms of gene therapies, again, this looks like a lot of words, but I've tried to help to break it down so that at a glance it doesn't look so overwhelming. So here, if a gene has got something wrong with it, or it's not there, you can replace it. You can give a vaccination in the form of bacterial or viral genes, which you will learn more about in Dr. Megan Lloyd's lecture on immunovaccination. Yes. And we have oncolytic microbes. Now, with this section here, it's a whole bunch of stuff that genes do. So they can speed the destruction of cancer cells, which means they kill the cancer cells faster, revert the cancer cells back to normal, so from cancerous tumor causing to a normal cell, or they can impede tissue growth. Because with cancer you have an excessive proliferation of cells which is uncontrolled, even in the absence of stimulus. So if you stop that, you stop the tumor from getting bigger. And finally, you can stimulate healing of damaged tissue. And with this last section here, you can see it's that nucleic acids can repair genes, so somewhat similar to the first point here. And it can help to modulate gene expression. So if you had a gene that was upregulated, you could downregulate it so that it's back to more or less normal levels of gene expression. Now, okay, we have reached the crux of this summary series. So, with the antisense therapies, these antisense transcripts actually occur naturally and they contribute to normal cell regulation. And what they've done is they've manufactured synthetic versions of this. So the antisense oligonucleotides, or AOs, can be used for 3 different things. First, they can degrade your transcripts. They can block translation. And they can alter mRNA splicing. You can see here 3 names of exon dis 51, which is what Professor Fletcher developed. And we'll go through this last because, well, it has the most detail, I feel we need to remember. Yeah. So, first up, we've got your ODNs. Now, I'll leave you to read that yourself. But basically what they do is that they can downregulate the expression of genes. So, and I'll go through the process a bit later on, but let's just have a brief introduction to ODNs. So, they were first developed to downregulate gene expression, but this failed because the oligo backbone binds to tumor stroma, which means the ODN cannot reach your target cells. Now, how it works is that the AO anneals to the target RNA, and this induces your RNase H, and your RNase H is what degrades your target RNA, and this helps to downregulate your gene expression. Now, on to another form of something that can help with transcript degradation. This was illustrative, so I don't think we need to know it in too much detail, but for completeness sake, I'll just go over it briefly. Now, as I mentioned, this is another form of transcript degradation, and also, like ODNs, naturally occurring, and its mechanism of action is that it does gene silencing by your siRNA interference. So, if you're doing cancer pathology, I've included a minor reference here from Dr. Meehan's lecture. This is where this description is from, and you might notice that there's some discrepancies between what's in Professor Fletcher's lecture and Dr. Meehan's lecture, but since this was illustrative, I wouldn't be too bogged down by detail if I were you. So just understand the general concept of risks. Yeah. So your siRNA binds to your protein complexes to form your risks, which are your RNA-induced silencing complexes. So once these risks are formed, they will bind to your mRNA, and these are complementary to your siRNA in your risk. Now the binding interferes with translation, so you get a downregulation of gene expression. Okay, now she briefly mentions microRNA, that it has multiple applications such as blocking binding sites, but it's not mentioned much in this lecture, so don't worry too much about it.